When we start, I think we are doing how do organisms reproduce. Till where we have still asexual. And that's the last chapter left, right? Our environment last है ना? तो reproduction में hardly और 2 lectures लगेगा. We are talking about sexual. Sexual part is there. And after that, our environment. So Okay. Now, let us continue. We had last class started with the concept of reproduction.
30 September bioscience.
हाँ बोलो.
30 September science.
30th September. तो अगर 18 को एग्जाम शुरू हो रहा है और coming in between possible होगा आने
अगर एसएससी होगा
तो वो थोड़ा देख के बोलना that you plan, see, check your timetable, plan which are the days we have the lecture. वैसे तो 30th के पहले पोर्शन वाइंड अप हो ही जाएगा. So in case you find out that it is possible those Tuesdays to come, then after this chapter is over, if I am doing essential now, so another 3 lectures maximum. 3 lectures में this chapter is over, then our environment एक lecture. Basic things. It's all very basic. Only important things I will tell you what to study. And certain things only you need explanation. Otherwise, everything you know. Now, we talk about reproduction. We had already discussed the other day about what? DNA copying. No, DNA copying. What is DNA copying? We studied. It is a process in which the chromosome will increase in number. Chromosome doubles. A process of cell division in which the chromosome number becomes double. And then the cell divides. And now, when we are talking about that the chromosome number is becoming double and the cell is dividing, during this process of dividing of the or division of the chromosomes, and later the doubling up of the DNA also, since this is a biochemical method, what I told you is there may be errors. As a result of the errors, the new cells which are getting those DNA are the changed DNA. Not like the parents exactly. So the DNA are the changed DNA. This changed DNA result in the formation of new characteristics that the parent was not having. What do we call these new characteristics? As variation. So this is what we had studied. These new characteristics are known as variation. Now variations are good or bad? Every organism, I talk about sexually reproducing organism right from the fishes to the mammals. Every organism is born with variation. Are we benefited by this method? Yes, because variation is making us more adapted, suitably adapted to the changing environment. Why some of these are absent today? This girl is absent. Exams. No. एग्जाम तो स्टार्ट नहीं हुआ. दिस लाइट कहाँ गए सब लोग? क्यों एब्सेंट? हाँ क्यों? एग्जाम्स 29 से एग्जाम्स हैं. टर्म एग्जाम है. Okay, now so when we are talking about this very what happened? एग्जाम्स है तुम लोग अभी कब से? हाँ. So tomorrow. पढ़ना नहीं था. हो गया पढ़ा. So now when we are talking about variation, now try understanding. Let me just give you certain examples. Any type of a question you should be able to answer. So when we are talking about variation, what is variation? Small little changes that an organism has as compared to parents. Why? Because the DNA gets changed. That's the reason. Now next is variation good or bad? That is what I was telling you. Variation good hai? हाँ, because variations are always making us better adapted to the changing environment as compared to our parents. I would talk about a parent bird gives rise to new generation birds, and these new generation birds develop, and these are semi-aquatic birds, for example, like the duck and the pelican, etc. Now, these new generation birds develop a longer beak. Beak thoda long hai. The next generation bird also, the beak is little bit longer. The next generation bird also, the beak is little bit longer. Is it an advantageous variation for these type of birds? Yes, because they can catch hold of the aquatic organisms and grab and eat. So it's a good variation. The other side, I talk about another species of birds, birds which take long flights now. So another set of birds, another species of birds. Now the new generation progressively, it is found that they are having the wings little bit more spread out. The wings little bit more spread out. Now it is seen that in almost about the new generation birds, in almost about 50 of them, majority of them have those wings spread out. Only 2 of them have got wings which are short. Only 2 of them out of 50 are known to have wings which are short. So what do I conclude from that? That the shorter wing is generally not is a disadvantageous variation. So this bird with that short wing, will it be able to take long flights, grab the food, protect itself? No. So quite a number of times it will be found that this bird is not able to survive for a long period of time, maybe not survive till the reproductive age. Now that it is not able to survive till the reproductive age, is it possible that this characteristic get passed on to the next generation? No. So this disadvantageous variation, the birds with the disadvantageous variation, are they generally passing it all to a large number of offspring? No, because they are not surviving only for long. Is this type of a story happening in the human also? Yes, when we talk about human also, out of 50 births, one is with a genetic disorder. Out of 50 births, 2 may be with some inborn diseases of the lungs and the heart and the liver. So these individuals who are known to have variations at a larger scale. Now the variation is at a larger scale and the variation is bringing about some disadvantages for them, not making them better suited. So how do we understand whether adaptations, whether variation is for good or for bad? If the variation is not allowing an organism to survive till the reproductive age, definitely that variation is a disadvantageous or a bad variation. And that is always in a lesser number of individuals. जैसे मैंने बोला अभी कि out of 50 birds, 48 was having that long spread out wings and only 2 were having the short wings. So any type of variation which is found in few organisms of a species always is understood to be a disadvantageous variation, and जो common advantageous variation होता है वो always spread out होता है because it is passing on generation and spreading out. So that is about the concept of variations. Now, variations if small, not a problem. We all are born with variations, but variations if big enough, major part of the DNA lost, major part of the DNA not copied, etc., then this is what is relating to abnormal or genetic abnormalities. This is what will be relating to genetic abnormality. So that is a basic concept of variation that you have to study. Next, we move on to a concept of the modes of reproduction. What are the modes now? The modes are 2 major, asexual and sexual modes. We will start with a table that we write. Differences between sexual and asexual, and after that, what are the differences you are not understanding, you will tell me. Take a table there, differences between asexual and sexual modes. Write down in your notebook, make it fast. Differences between the asexual and the sexual modes. Make a table, asexual reproduction and sexual reproduction. Asexual and sexual reproduction. Asexual and sexual reproduction. Under asexual, you write. Under asexual, only one parent is involved. Under asexual, only one parent is involved. Under asexual, only one parent is involved. Under sexual, two parents are involved. Two parents are involved in the process of formation. Two parents are involved in the process of formation of an offspring. In the process of formation of an offspring. Number 2, for asexual, reproductive cells known as gametes. Reproductive cells known as gametes are not formed. Reproductive cells known as gametes are not formed. Other side, sexual, gametes are formed. Gametes are formed. Number 3, asexual. Offsprings formed are many. Offsprings formed are many. Offsprings formed are many. Sexual. Offsprings formed are few. Offsprings formed are few. Next. Asexual. Offsprings formed are identical. Offsprings formed are identical. Sexual. Offsprings always show variations. Offsprings always show variations. Now, this few points of difference is always to know. So, what are we now summarizing? If it is a concept of asexual reproduction, I may talk about the amoeba, I may talk about the hydra, I may talk about the spirogyra. In all these organisms, what is happening? The organism is giving rise to new generation organisms, many at a time. And all the organisms are identical and we are not able to find out any differences between them. Now, can I say that there are variations? There are, but it is negligible because it is the DNA of only one parent involved. But the moment we talk about sexual reproduction, even for a fish or even for a amphibian, though we don't understand those differences there also, but in close study, if we study those organisms closely, we will be able to find out differences between the parents and the offspring. So roughly to understand this way right now, that if it is a concept of sexual reproduction, the offspring will be always formed with the help of two organisms, and if it is asexual, only one. There is nothing known as specialized cells or gametes in case of a asexual method, and gametes is a must when we are talking about the sexual method. Offsprings formed in the asexual method may be numerous, even in hundreds at times. And if I talk about the sexual method, it is a more complicated, complex, lengthy process, so the offsprings formed are always much lesser. Now let us start talking about some of the major asexual modes. What are some of the major asexual modes that we start with? We are starting with a process known as fission. We start with process known as fission. What are some of the differences, the major characteristics? Let us try understanding. Fission, when I start with, the first method of fission would be binary fission. Binary fission. Now, binary fission, what type of organisms and what would be the method? Binary fission, I would say, A specific type of an organism, this organism shows changes like elongation of the nucleus. These diagrams are very important for you. They are simple, very simple, but definitely one diagram drawing will definitely be there in your paper. And description, 2 mark, 3 mark. So try to pick up marks for these type of questions. You don't have to think too much to write. Very easy, straight questions. So when we are talking about binary fission, what is binary fission? I talk about one single-celled organism. I may talk about the parent organism, single-celled organism. So what is that change? First step is elongation of the nucleus. The first step is what? Elongation of the nucleus, and this is followed by the division of the nucleus where the nuclear constriction takes place. So next step would be the nuclear constriction. How many times the nucleus is dividing? Just once. The nucleus is dividing once. And then this would result in the development of 2 daughter cells, and here we are talking about the daughter organism. So they can also be written this way, the daughter MEV. What is this method? What are the changes? Just see here. So simple here. We are talking about the parent organism. The nucleus gets elongated, followed by the constriction of the nucleus, and this would be followed by the division, the constriction of the cytoplasm later, and the 2 cells are apart. This is called binary fission. But what is the contrast? The other method, when we are talking about fission, there is one more method of fission. What type of organisms for binary fission? You may talk about organisms like amoeba, you may talk about organisms like the paramecium, organisms like the euglena, all these different type of organisms are in this category. Binary fission is this. The second method that we talk about would be multiple fission. How is multiple fission different from binary fission? Remember what points I speak. So when I give you the answers to write, you should be able to write down the differences. Multiple fission. Now when I, yes.
Binary fission only 2
daughters. Always.
and multiple and there are multiple.
Many. There are something more. There is something more also to find out. So now that I talk about that these type of organisms, I may talk about one parent organism. What type of examples? Basically, to understand examples for multiple fission, example I may write Plasmodium. Generally, we come across disease-causing organisms generally show this method. Disease-causing organisms generally show this method, and free-living organisms generally show this method. Free-living meaning organisms which are not capable of causing any harm, no diseases actually. So I would say that this is the parent Plasmodium. This is the parent Plasmodium. What is happening to this organism? It is undergoing nuclear division, and the nuclear division a number of times now. Multiple nuclear division it would undergo. Multiple nuclei it would generally form. So as a result, now this Plasmodium is forming this many nuclei. So many nuclei it would generally form. And after a particular period of time, every single nuclear fragment generally gets wrapped up by a small amount of cytoplasm, something like this, generally gets wrapped up by a small amount of cytoplasm like this, resulting in the formation of a cell-like structure. Resulting in the formation of a cell-like structure, something like this. And later, this cell-like structures all, once they fill up the entire space of the cell and the membrane not able to hold it anymore, the entire membrane will result in breakage, rupturing that we understand, and this daughter organisms are all set free. The daughter organisms are all set free. Now this is the new organism, the new generation organism, so many at a time that we understand. And I also mentioned multiple fission is generally for organisms which are capable of causing diseases also. Now I picked up this example, Plasmodium, which is capable of causing malaria, capable of causing malaria. So these are the daughter nuclei. These are the new daughter organisms. These are the daughter plasmodium, so many in number. So this is a major difference when I talk about the binary and the multiple fission. What are some of the differences that you write? Here nuclear division only once, here nuclear division multiple number of times. Here two daughter cells formed, multiple daughter organisms will be formed. That is majorly for pathogenic organisms, disease causing organisms. Majorly for pathogenic organisms, and this side when we write majorly for pathogenic organisms, and this side when we write what it is, free living organism. They are generally not capable of causing any harm or any disease.
In the third step, multiple it's getting wrapped
By a small amount of cytoplasm.
which
Then once they are getting wrapped by a small amount of cytoplasm, now this is almost the formation of the new generation organism. Now they are almost filling up, membrane burst, and they are free. Now something more that is here is that every single organism, here now that we understand, every single organism, they may be additionally covered and protected by some cyst wall. They may be additionally covered and protected by some cyst wall. What is the function of the cyst wall? Avoid unfavorable conditions. To avoid unfavorable conditions. So now these organisms, that's the reason immediately they don't die out. They may be released in an unfavorable condition, too much of dryness or too much of high temperature, but immediately are they going to die out? No, they will not die out because that hard covering will allow them resist the environmental laws for a particular period of time. What about this side? Nothing as such like this. If this new amoeba developing in a unfavorable condition, they die out, right? So this is the major difference, 2-3 differences that I spoke about, and the organisms which show this method. This is what to write for binary and multiple fission.
explain the multiple fission.
Multiple fission, if it is a parent cell will show multiple times the nuclear division. The nuclear break up into a number of fragments. Then once an appreciable number of fragments are formed, the cytoplasm, the cell has got the cytoplasm also, yes or no? This cytoplasm will break up into multiple fragments, and every smaller amount of the nucleus is getting wrapped up by a small amount of cytoplasm as well.
Once the nucleus is getting
Wrapped up by a small amount of cytoplasm. Every single nuclear fragment wrapped up by a small amount of cytoplasm. That's how the cell is getting formed. The new organism is getting formed. But this new organism is getting matured only once it is released from the breakup from the membrane and release. But at that time, if it comes in contact with some unfavorable conditions, if it comes in contact with the unfavorable conditions, it will immediately not undergo the process of growth and maturation. It will hold for a particular period of time as long as the situation is favorable. So this is what you write, differences. Now tell me one similarity between these methods. A name. One similarity between both these methods. After the new generation organism is formed, do we get to see the parent anymore?
No.
So we would write, what is that similarity we write? Parental identity is lost in both the methods. Parental identity is lost in both the methods. That is the similarity always to understand. Now, next is, after the concept of fission, we come across a concept known as fragmentation. What is fragmentation? Fragmentation majorly happens in multicellular organisms, but still they are simple. They are not that complex multicellular, we should write. They are simple multicellular organisms. Example like Spirogyra, example like Lobelia. These type of organisms which are present in the shallow waters, shallow water body. Now, this type of organism generally has body something like this. They would generally have multicellular body, just now as I mentioned, but then the body is not very complex. It's not a complex structure actually. simple structure they would have. And what is a specialized feature that they have is a ribbon-like body, and then they have something known as similar, like the DNA, the hereditary structure, and then the nucleus there, and a small bit of cytoplasm. That is their cellular structure, nothing much that they have. The cellular components are quite simple. Some strings of protein, some amount of enzymes, and then the nucleus, and then the cellular structure. So what is this filamentous structure known as? This filamentous structure is known as the thallus. The entire mature body of the organism, the filamentous ribbon-like body of the organism in the branching pattern, इसको बोलते हैं thallus. Now, this parent organism, so I would say this is the parent Spirogyra. What happens to this parent Spirogyra when absolutely mature, when this organism is absolutely mature? What would happen? The body divides, the thallus divides. What happens to this organism? The thallus normally, naturally divides into certain parts. The thallus will normally, naturally divide into certain parts. So I would say that this thallus is actually dividing into certain fragments. The thallus is dividing into certain fragments. Now, as the thallus divide into certain fragments, we are going to come across that how many daughter cells to come out from this. How many daughter cells should come out? That would very much depend on every particular fragment. Now, I would say that this is dividing into a number of fragments, something like this, and maybe something like this. So how many fragments am I talking about? I'm talking about 1, and then 2, and then 3, and then 4 fragments. So how many new Spirogyra expected from here? 1, 2, and 3. So now, no, not 4. This will not give rise to a new Spirogyra. Now, what is the reason to write? When the entire thallus is divided into a number of segments, every segment, do they have adequate number of cells? Do they have adequate number of cells? Now, I already mentioned this is a simple but multicellular structure. Do they have adequate number of cells to multiply and form the new organism? I talk about this fragment, I would say no, this will never give rise, because a well-developed cell is not there at all. It's a absolutely broken bit. So this cannot give rise to a new organism. So how many organisms I come up from here? 3 Spirogyra we would generally come up from here. So in your diagram, in your question paper also, you get sometimes diagrams like this and certain questions. So see, try understanding and answering properly. So now here, how many offsprings? 3 offsprings, all are identical. 3 offsprings, all are identical. Now that I talk about all these 3, organisms, is the parent there parallelly? No, the parent is not there parallel. So what do I write? Is the parental identity retained or the parental identity lost? The parental identity is lost. Here also, the parental identity is lost. So how many Spirogyra we generally get here? We get here 3 Spirogyra. Don't do that. Sitting in the first bench, do you want me to yell, shout at you? I don't want to do that. 10th standard ko kaun डांटता है? Doesn't look good. Don't do that. Don't give me chances, क्योंकि मैं एक बार जब अपने मूड पे आ जाती हूँ, then I will not spare anybody. So don't instigate. So when we are talking about this Spirogyra, so can you tell me now a difference between fission and fragmentation? One difference between fission and fragmentation. Parent was giving rise to only 2 offspring. That was standard. 2 offsprings. 2 से 3 कभी हो सकता है? No. In case of fragmentation, how many offsprings will be formed? Based on how many fragments the thallus divides into. यहाँ पे कोई standard 3, 4, 5 कुछ नहीं है. I would have shown a diagram something like this. अगर ऐसा है तो this part will give rise to 1. This part also will give rise to 1. Adequate number of 2-4 cell है तो it will give rise to 1 filament. Absolutely कुछ भी cell नहीं है, then it cannot give. So how many offsprings by this method depends on the number of fragments. How many offspring by the process of fragmentation depends on the number of fragments, which has adequate number of cells. That will definitely give rise to a particular fragment there, to a particular things. So this is about fragmentation. What type of organisms show fragmentation? Organisms like Spirogyra, etc. Next, the fourth method that we talk about is budding. The fourth method, the third, no, we are talking about the third method. The third method that we talk about is budding. Now, first of all, if I talk about budding, budding of 2-3 types. First is budding in unicellular organisms. Budding in unicellular organism, what type of organism I'm talking about? One is the yeast, the parent yeast, I may understand. What would happen? This yeast would generally result in the formation of a bud. एक bud है, a small bud starts getting formed. Initially, a bud only starts getting formed. Then next word, this parent organism will result in the formation of the bud, and the nuclear division has already taken place. Later, this bud will result into a formation of a complete new organism, a fully developed organism. This parent is still there. The bud is fully formed, and this bud has now given rise to another small bud. So it is like this, the method. Which type of organism I'm talking about? The yeast. Where do we use the yeast? Where did you come across? Did you ever come across the usage of yeast? In the process of fermentation or baking and all these processes. So when we are talking about a chain of buds generally get formed this way. Now what is one difference in the process that we talk about? If it is a parent organism, this is what is a young bud form. This is a young bud form. Then the bud grows and remains attached to the parent organism and remains attached to the parent organism. Then once it remains attached to the parent organism and once it is fully grown, does it get detached? No, it does not get detached. Then it will again give rise to another bud and the process continues. But do they get detached? Yes, after long chains are formed, then abruptly they will detach. There is no programmed separation from the parent cell. Abruptly they detach. So that's the reason whenever we get to see this, there's fine filaments also of the yeast, we definitely get to see them with our naked eyes. We can see, but that is actually a number of those fine thread-like structures, a chain of buds, a number of them together. That is what we get to see as threads. Put them under the microscope, it's actually not one thread, hundreds of them are together. This is budding in single-celled organism. Remember the examples also. That is budding in single-celled organism. Then what about the budding in multicellular organism like Hydra? Budding in multicellular organism like the Hydra. Now budding in multicellular organism, if I'm talking about, that would be in case of some slender cylinderates. Hydra is a cylinderator. So this parent Hydra, how will it start the process of reproduction? Results in the formation of a bud like this. Results in the formation of a bud like this. Now the development of the bud, anything programmed? Any definite number is there? Nothing. The formation of the reproductive cells here does not mean gametes. The specialized cells which undergo the process of budding. These specialized cells aggregate and result in the formation of the bud. Now the bud for one parent, maybe 2, maybe 8, maybe 10, depends time to time. There's nothing specific here. So these organisms, let us try understanding talking with 2. Then what would happen to this organism? This organism now will show the process of development and the organism is growing. What about this organism? Yes, this organism is growing now. This bud is constantly in the process of growth. When will it get detached? It will get detached. Is it like this, remaining attached? No, it will get detached once it is fully grown. So this hydra, once it is fully grown, it will start detaching the new generation organisms. The new generation organisms gets detached from the parent organism once they are fully grown. So here we talk about the daughter hydra. Here we talk about the daughter hydra. Tell me now one difference between this method and fragmentation.
Parental identity.
Here, these are the new generation organisms and the parents still there, no? So what is this feature known as? This is, you will have to write differences like this. The parental identity is maintained or parental identity is retained. It is still there. The parent is still there. In case of fragmentation, no. Parental identity is not there. Lost. In case of fission, lost. So budding is one such method where we understand the parental identity is maintained. So that is about budding. What are the two types of budding we came across? In case of single cell organisms, how it is. In case of multicellular organisms, simple multicellular organisms, how it is. The two different types of budding. What next? We talk about spore formation. We talk about spore formation. Now, what is spore formation and where does this take place? In case of spore formation, majorly we are talking about organisms like the fungus. Variety of fungus show this method. Examples what? Examples like the mucor, examples like the bread mold, examples like the rhizopus. All these show variety of methods, all these variety of fungal organisms. Where do we see them? Growing on the food items, growing on the leather items, growing on the damp and the moist surfaces. How would the method be? This type of organisms, they have got a slender body, something like this. They have got a slender, and this is the multicellular slender body. What do we call this as? This is known as the hypha. Multicellular slender body, multicellular structure. Now, this type of multicellular structure we often get to see, especially in the moist weather, on the surface of the bread, on the surface of the fruits, on the surface of the cooked food. If it is kept outside for maybe 1 or 2 days, fungal infection in this weather very fast. So now on this stale food, decomposing food, the bread as I said, or the fruit as I said, when it is overripe, we get to see a cottony growth there. Now that is what are these fine cellular structures, fine hair-like processes. Like this is what is known as the hyphae. Now next one, these type of structures, they will generally show growth something like this. One of these will start showing a particular process of growth. Now, that is typical cell division to understand. So what is happening here? One such group of cell. Now, in each of these hypha, it is just one cell which has started showing multiple nuclear division, something quite similar like the multiple fission we were talking about, where nucleus dividing many number of times. Then what? What are these structures known as? These structures are known as the sporangium. What are these known as? These are known as the spores. And what are these now known as? This is known as the sporangiophore. So now that I talk about the sporangiophore, तुमको सब आता है क्या? हाँ? तब तो मैं रेस्ट करती हूँ, मैं क्यों इतना बोल रही हूँ? मैं थोड़ा रेस्ट करती हूँ ना 10 minutes. So now when we are talking about this, the fine hair-like structures, these fine hair-like structures, what are they known as? Hypha. Now when this hypha gets matured, it is not the entire cell, the entire structure does not show multiple division of the nucleus, it's only one cell which will show multiple division of the nucleus. And now The nucleus results in the formation of a large number of nuclei. So this nuclei we rather give a specific term. What are these specifically known as? Spores now. Now here, unlike multiple fission, in case of multiple fission, what did we see? Every single nucleus is getting wrapped up by the cytoplasm. Here, not that much because the amount of cytoplasm is as it is so scanty there. So it is not that a process. But later, what will happen is once the sporangium is quite mature, what now later is this sporangium will start rupturing up. This sporangium will start rupturing. So I would say that something like this sporangium starts rupturing and a large number of these spores are dispersed. A large number of these spores are dispersed here. So a large number of spores are released and they are carried by the wind to long distances, and this is one reason why the fungal infection happens so fast. Fungal infection is generally uncontrollable because the spores are very light, very small, numerous, and they can travel long distances, and they're also very fast. So when we are talking about the process of spore formation, a type of a division, a sexual mode of reproduction in case of the fungus, where the rupturing and the breaking up of the sporangium will release millions of such type of sperms. These spores, and these spores now under favorable condition. Again, I'm talking about similar like the concept of multiple fission. Provided they get the favorable condition, they will grow in large numbers. Then, if they don't get the favorable condition, they may remain dormant for a long period. They may remain dormant for a long period with the help of a cyst wall. In case of fungus, that is very, very common. This small pores with the help of a cyst wall. What is this cyst wall made up of? Majorly a coat of protein. And it can protect the spores from any type of environmental odds. Very high humidity, low temperature, high temperature, etc. And now these spores get favorable conditions and they will start germinating again. Yes. The release. I said that once we are talking about this hypha, now what will happen? Now that the fine hair-like structures have started growing, now that they start growing and the humidity and the temperature is still favorable for their growth, then one of these cells will start multiple nuclear division. And that's the reason I think quite a number of you must have noticed that also on the surface of the bread. Today you get to see some fine cottony hair growth. You leave this bread, tomorrow you get to see some black spots or some green spots at the top. What are these green spots or the black spots at the top? These are those sporangia. So if this slice of bread you keep for another 2-3 days, the formation of the sporangium, we can see with the naked eyes those black spots there. And this is the sporangium with numerous spores there. After some time, what will happen? They will burst out. Spores release. And since the spores are so very light, so very small, easily they can be carried long distances. So the fungal infection can spread out very fast. You talk about fungal infection not only on the bread, you talk about fungal infection on the scalp, on the skin. Very difficult to treat because it spreads from one region to the other very fast, right? So that is about spore formation. Now these are the typical methods of asexual reproduction. What are the ones I spoke about? Fission, fragmentation, budding, and spore formation. What is left is regeneration. Let's talk about this. We talk about the next method, that is regeneration. Yes, सारे methods clear है? Now we talk about the next method, that is regeneration. Now how is regeneration different from this method? Regeneration is not actually a method of asexual reproduction, but it is actually a method of regaining the lost parts. A method of regaining the lost parts of the body. Regaining the lost parts of the body. The lost parts of the body. Now, if I talk about any particular type of an organism, I talk about any particular type of an organism. Now, say suppose I talk about the planaria. Now this planaria that I understand, this planaria that I'm talking about, it is a flatworm. This planaria is a flatworm. Now this planaria, we are saying that the planaria body divided into three parts. And we are saying that this three parts that the planaria is divided into, this should give rise to a new organism. This should give rise to a new organism, and this should give rise to a new organism. Good enough. Yes, if the body divides into three fragments like this, of course we are going to get three new planaria. Again, the concept remains the same. If I would have shown a fragment this way, then this perishes up. So that is one concept. The second thing is the planaria like the Spirogyra? Is the planaria like the Spirogyra dividing its body naturally? No. Because the planaria has got its own method of binary fission, which is known as transverse binary fission, obliquely hota hai. So this particular planaria, by as accident in the water body, if its body gets divided. Now, as a possibility hai, not necessarily every time the planaria's body should get divided like this. It is also possible that this flatworm, the body of this flatworm divides almost like this. The body of the flatworm divides abruptly like this. And how many planaria we are expecting? Three yahan se bhi, or maybe now four from here, as long as adequate number of cells are there. But is it all planned like the Spirogyra? No. It is also possible that the part of the planaria is all been eaten up. The part of the planaria is all been eaten up by some other different aquatic organisms. By accident, always their body is broken. This type of organisms, they never break their body naturally, but only by accidents. Their body generally get broken down. But a speciality that they have is by simple cell division, a new planaria may completely get formed. By simple cell division, क्योंकि इसकी body by another organism has eaten a part of its body. So अभी इतना सा ही बचा है. This can give rise to a new planaria. कैसे? It does not have any well-developed tissues and organs. So by simple cell division, the entire body can be revived back. But I talk about a frog. I talk about a lizard. I talk about a frog. I talk about these vertebrates. Do they also undergo regeneration? Yes. But talk about this lizard. This particular lizard has lost almost a part of her tail, big portion of the tail. Now you get to see that this lizard is moving around out there, and it is having only a small portion of the tail. You get to see the lizard, one particular limb not there. Out of the two pairs of limbs, it has only three limbs, one limb not there. Now, what is happening? Will the lost limb be formed in this lizard's body? Will the lost limb be formed? Yes. But वो limb से नया reptile बनेगा, नया lizard बनेगा? No. Why not? That's a question you get in your paper. Ki if I talk about this entire lizard and the lizard has lost its arm, what is the probability? What is the possibility that a new organism will be formed? No new organism will be formed. Only that the lizard's body will regain the lost limb, but the lost limb will just decompose. That will just perish away. Reason क्या है? वो lost limb से पूरा एक छोटा सा reptile lizard क्यों नहीं बनेगा? Because it is a vertebrate. The entire process of development of the tissues and organs, right, starts from the embryonic stage. And at any time of life, our body, vertebrate body, cannot give rise to any tissues and systems because they have got well-developed systems. इसमें तो कोई system नहीं है, तो इसलिए problem नहीं था. Few cells by mitotic cell division and the body. That's it. तो इसमें क्यों नहीं हो सकता? Talk about the frogs. Talk about the frogs. Now that I talk about the tadpole, now that I talk about the adult frog, lost one limb, lost two limbs by an accident. What would happen? Can the limbs again revive to form the new body? Of course not. And to some extent, the frog's body can revive back the limbs, but फिर भी इतना efficiently नहीं होता. You talk about human also who has gone through amputation or who has gone through loss of the fingers and the toes and the leg. What happens? The body can to some extent regrow and repair. पूरा limb बनता भी नहीं है. पूरा limb क्यों नहीं बनता है? Because the entire process of development of the tissues and the organs embryonic stage से ही develop होता है in case of vertebrates. Any time during the life cycle नहीं हो सकता because we have complex tissues and organs. So right, any time of the life जब आधे से ज़्यादा tissues बन चुका है, suddenly one particular tissue cannot be formed. Does the human body undergo the process of regeneration? Yes, the liver undergoes regeneration. A person who has gone through liver surgery, a person who has gone through liver transplants, yes, regeneration happens, but if a person has gone through liver surgeries, 50% of the liver cut out, 40% of the liver cut out, वो पूरा liver बनेगा? No, 20% ही बन पाएगा. So what are we understanding? What is the concept of regeneration? Does the planaria otherwise have any method of reproduction? Definitely. Planaria has another method of reproduction because this also has to continue its race. But by chance, if it suffers from an accident, then it has got a simple method of division to form its entire body. Upper body lost hai to lower will form the upper. Lower body lost hai to upper will form the lower. So this can happen. Give reason. Planaria can form complete organisms by regeneration, but a lizard cannot. Are you able to write down the reason now? Yes, because they don't have complex tissues and systems, and by simple method of cell division, the entire organism can be formed. But this group of organisms, vertebrates, have got complex systems and organs, and the entire process of organ systems starts from embryonic stage and follows a definite pattern. So that cannot happen at any stage of the life, and with various examples. So this is what we are talking about as a concept of asexual reproduction. Next, what is vegetative propagation? Let us start with that. What is vegetative propagation? Vegetative propagation is actually a concept of asexual reproduction in plants to mention. Vegetative propagation. What is the meaning of propagation? Propagation मतलब नहीं पता. Vegetative propagation. When we are talking about a concept of vegetative propagation, propagation means developing and multiplying in large numbers. Vegetative, non-reproductive. So which are some of the non-reproductive parts of a plant? Now we start understanding this. What are some of the non-reproductive parts of a plant? The non-reproductive parts of a plant would be the stem. The non-reproductive parts of a plant would be the root. The non-reproductive parts of a plant would be the leaf, the bud. Now, this particular method, where from these parts of the plant, a new plant can develop. If I am talking about a method by which these parts of the plant can give rise to a new plant, that is what is vegetative propagation. And what is the definition? The process by which non-reproductive parts of a plant like this can give rise to a new plant. Write down. What is the method, the definition, and the differences you should know. What is meant by vegetative propagation? The method by which vegetative or non-reproductive parts of a plant. Method by which vegetative or non-reproductive parts of a plant, vegetative or non-reproductive parts of a plant give rise to a new plant. Give rise to a new plant. Give rise to a new plant is known as vegetative propagation. Now, try understanding. अब ये जो method है vegetative propagation, in this method will there be involvement of any reproductive cells? No. It's a quite similar like a sexual method we are talking about. So now the plants formed by this method, should they be all identical? Yes. Can we grow large number of plants by this method? Yes. Vegetative propagation hai. So I may talk about any particular plant. Now say suppose this particular plant has the stem and I try taking stem cuttings. I try taking stem cuttings. Now this is one stem cutting that I get. The stem cutting has to be something roughly around 3 to 5 centimeter का होना ही चाहिए. The stem cutting that is minimally required to grow any particular type of a plant, 3 to 4 centimeter, 3 to 5 centimeter minimum होना चाहिए, and the cut should always be oblique. If anybody has noticed that the cut for the stem cannot be, the cut always has to be oblique. Can you tell me why? If anybody has seen the gardeners जब काटते हैं, they don't cut it this way, they always slash it this way. And वो जो stem cutting होता है, that always has to be always obliquely होना चाहिए. क्यों? Any idea? Now that I put this inside the soil, say suppose now that I put this inside the soil, it has got enough surface area. More number of cells are exposed to the soil water. Absorption of water is even more. Absorption of nutrients even more. And the process of development, cell division much faster. As compared to a stem which I put this way. As compared to a stem which I put this way, the surface area is much lesser. So why should the stem be cut obliquely? The stem cuttings, why should they be obliquely while we put them in the soil and try growing a new plant? Your answer always should be based on this concept. So the surface area is more, absorption of the water nutrients more, and fast development of the root hairs. So अब जब root hairs फटाफट develop हो जाएगा, the plant is already established and the growth of the shoot out there you will understand. So ये हो गया first concept of cutting. Cutting is one method of vegetative propagation. What type of stems can be grown by cutting? A variety of roses, variety of China roses, variety of roses, various different type of sugar cane. So quite a number of these plants which have got developed stems. Cut them, slash them obliquely, put them in the soil. If the soil has got adequate amount of water, nutrients, definitely they should be growing. This is one method of vegetative propagation. The next class will start with layering and grafting.
इनाया को नहीं दोगे, इसको लगेगा मैं कुछ फिक्स करके जीरा दिया